In this project, we will illustrate the molecular mechanism of HIV infection, how it affects the immune system, and current drug studies being performed to alleviate the deleterious effects of HIV. HIV, or human immunodeficiency virus, is a retrovirus that hijacks the host cell's transcriptional and translational machinery upon infection, converting its single-strand positive sense RNA into cDNA. HIV also integrates itself into important immune system cells and disrupts their function, inhibiting an organism's ability to fight infection. HIV has also been known to mutate very quickly, so it's very hard to control and find a permanent solution. To infect host cells, HIV contains two glycoproteins, GP41 and GP120, which bind to the CD4 receptor of host cells and mediate its fusion with the plasma membrane. This fusion deposits HIV's single-stranded RNA genome, along with other various proteins such as reverse transcriptase and integrase, into the cytosol of the host cell. In the host cell, reverse transcriptase converts the viral RNA to cDNA, which is represented here by the pink strands. The cDNA, other viral proteins, and integrase combine together to form a pre-integration complex called the PIC, which then enters into the nucleus. Once in the nucleus, integrase creates a double-stranded DNA break that allows for integration of the viral cDNA into the host genome. Research published in 2002 by the Infectious Disease Laboratory at the Salk Institute in California demonstrated that HIV's integrase enzyme exhibits a bias to where the new cDNA is integrated into the genome. The author, who infected a human T-cell line with HIV and then isolated the DNA from these cells, sequenced these strands and found that a majority of HIV cDNA was integrated immediately downstream of genes that become activated during infection, locations which he termed hotspots. This, they concluded, contributes to HIV's ability to immediately transcribe and translate its newly inserted cDNA using the host cell's machinery, causing the cell to generate new viral RNA and proteins for production and assembly. When the HIV virus has completed its replication cycle within the host T cell, it stimulates the tumor necrosive factor that mediates apoptosis of the host cell, allowing new viruses to be released to infect more cells. This constant apoptosis of T cells causes the characteristic low T cell count among AIDS patients, of which HIV is the causative agent. So how could we potentially stop HIV? Since the viral RNA is constantly mutating and changing, it is difficult to target with inhibitory drugs. There are, however, several current inhibitory drugs that target the CD4 receptor, plasma membrane fusion, reverse transcription of viral RNA to cDNA, and integration of viral cDNA into the host cell genome. So read your perusal, and maybe you can be the one to finally find the cure for HIV. I got loyalty, got royalty inside my DNA. Quarter piece, got war and peace inside my DNA. I got power, poison, pain, and joy inside my DNA. I got hustle, though ambition flow inside my DNA. I was born like this and sworn like this.